thank you. Thank you for reaching out. Thank you for having me. And so today I'll be talking about tools for bacterial analysis and how they really show disease transmission and evolution. And to go through that, I'll be talking about public health and genomic epidemiology, the role of bacteria in healthcare, the typical workflow for bacterial surveillance, limitations to current approaches, and then the background on software tools for bacterial data analysis and population sequencing, specifically with looking at two different studies with the baby biome and the nursing home. And so public health epidemiology is really how diseases occur in different groups of people and understanding their patterns and methods and preventions. And it's really a crucial role in monitoring outbreaks and communicating information to healthcare professionals, right? And lay people too, and thinking about how to minimize the harm done to the larger population. And so public health was really brought to the forefront during the COVID pandemic. And it was standard to see people take nasal swabs or to have saliva samples. And there were multiple layers as to why this happened. The main purpose of swabbing was to determine if people were positive or negative, right, for COVID. Do they have COVID? And you can start to answer different questions about who is more at risk. So they were able to compile the information and create databases at the national, regional, or even institutional level, as was done at UN Hospital. And they wanted to see how prevalent was COVID across these different layers and if there was greater patterns about where COVID was occurring as opposed to other places. Then genomic epidemiology came into play and sort of the background. And it's about studying the specific pathogen genomes in order to really gain a deeper resolution on transmission dynamics. And so you're asking questions like, does everyone have the same type of COVID? Do different strains have different levels of transmission? And do strains cause more severe disease than others? And so these are really important questions that you can't answer without genomics. And so I've been talking about COVID, and one person in my lab does COVID, but I do superbugs, which are strains of bacteria which have evolved to resist antibiotics. And the widespread use of these antibiotics in the 1900s is what really caused this and was a big catalyst for this. And so while COVID is a virus, how we study drug-resistant superbugs is sort of can be the same and overlap in many ways. And so some factors that contribute to antibiotic resistance include like the overuse and misuse of antibiotics, the unregulated use of antibiotics, and humans in healthcare and in an agricultural setting. And so some organisms that I work with are MRSA and VRE, methicillin-resistant Staphylococcus aureus, and vancomycin-resistant Enterococcus. These are two big organisms that are very prevalent in Michigan hospitals. And so MRSA is really the most deadly pathogen associated with bacterial infections. And so these pathogens are multi-drug resistant, which means, again, they've evolved the ability to bypass several drugs designed to treat them. And although people can carry these bacteria and be completely fine, immunosuppressed individuals like those with cancer or HIV are more likely to develop horrible infections. And then finally, we talk about the spread of disease. I can't leave out things like the hospital-acquired MRSA, first emerged in hospitals due to selective pressures, and then it adapted and spread beyond hospitals into what's known as community-associated MRSA. And the big thing about this is about 70% of hospital-acquired infections are preventable. And they're just unfortunately, more common than we'd like them to be. And so how do we go about treating this, right? So I talked about public health, epidemiology, and the role of bacteria in healthcare. And now we're going to get into a typical workflow for bacterial surveillance. And so sample collections first are swabs taken from different sites in the hospital and the patient's body, right? We talked about nasal swabs, saliva. When you're dealing with certain bacteria in the hospital, sometimes they're more prevalent in certain areas of the body. VRE, for example, resides in the intestine. So you would take a groin swab, MRSA, a nasal swab, and things like that. And so you collect these bacteria present at these different locations, and then you culture the selected samples, and you spread them and you incubate them. And then after you spread the collected samples on agar, you select, right, for specific species that you want to work with in order to really grow MRSA or resistant bacteria. And so this selection step really enriches the bacteria of interest, right? For example, specifically MRSA strains, are allowing researchers to really identify whether a sample is positive for the pathogen based on growth, color, and things like that. And then finally, you have the colony isolation, right, where you pick a colony that represents an individual proportion of a population, and you isolate it from the selected plates, and then you test this isolate, whether it's doing antimicrobial testing, susceptibility, or genetic analysis with Illumina sequencing. And so after sequencing these reads from each isolate, then they're aligned to a reference genome based on the species. And so in order to choose reference genomes that are particular lineages, there are particular lineages that are more commonly associated with particular regions in the US or particular regions throughout the world and in North America. So ST5 and ST8, just for example. And people typically base these lineages of bacteria off of sequence types, which is a set of housekeeping genes. And so you have these variants that are called and filtered, and then you develop SNP matrices that are then calculated between each genome for each various species. And so it's a pairwise comparison of regions that have variation across all of the genomes. And then only the variants that are located at nucleotide positions that are present in all of the isolates are considered for a transmission analysis. The thing about this is that it doesn't take into account that bacteria exist in mixed populations, right? Bacteria are not isolates. A bacteria strain, for all of our purposes, is a genetic variant of a bacterial species. And so these strains are groups of bacteria within the same species that have distinct genetic and phenotypic characteristics. They may differ in resistance profiles or metabolic capabilities. And so with the population of antibiotic-resistant bacteria, you can have many combinations and trajectories for phenotypes. Not every organism in a species may react the same to particular treatments. From one methicillin-susceptible Staphylococcus aureus, you can develop resistance to multiple drugs, which will then go into different epidemiological contexts of whether something is clinically associated or community associated. And so with this current approach, there's limited diversity being captured. Isolate sequencing typically examines a small number of colonies, which may really miss minority or co-colonizing strains present in a sample, really underestimating the true diversity that exists. You also have sampling bias, and so the choice of these colonies for sequencing introduces biases, as not all strains are equally likely to be chosen. Not all strains are likely to grow in the same conditions on the same media, really affecting the representatives of a population. And of course, higher costs and labor. You have to think about the people going in there, interrupting positions, swabbing areas like the nose, the groin. It can be really taxing on patients that have to do this over and over again and have multiple samples taken in one day. And then the lack of variant frequency data. And so with isolate sequencing, we cannot quantify allele frequencies or strain abundance within the original microbial population. And so we're able to do that analyzing the whole population as a whole. Then we can see how alleles shift over time and change. And then finally, oversimplified transmission inferences. And so because of this limited diversity representation, isolate sequencing at the end of the day may fail to really identify transmission links between people and communities. And so we talked about how, some microbes may not grow as well on the medium that you use. Some species may be faster growing as compared to others and outcompete them. And that may be a result in the downstream analysis. And then how people pick colonies leads to the opportunity to miss rarer or slowing rarer organisms. And so we talked about the limitations to current approaches. And so my first step was to search for bioinformatics tools that are good at detecting various strains of bacteria, search for tools that are able to separate the genetic differences between similar strains of bacteria in larger populations, and tools that could help me make inferences about transmission and combine different aspects of tools together. And so how did I do this? Like us all, I go to ChatGPT, I type in some stuff, and I hope it gives it back, right? Reading articles on PubMed, going to Google, GitHub, and having like 50 tabs open and doing this process over and over again. And so I came across so many different approaches and technologies, and my PI was like, make a list of these tools and papers and summaries and citations that'll help me later in writing papers in my dissertation. And of course, there are technologies to compile papers, right, and citations that already exist, and you can get papers automatically sent to you. But I was looking at things, there wasn't something out there that would compile the information for newly released tools and compile it in a way for me. And so I did use AI and Google and GitHub, but I realized I was making, again, the same searches over and over again. And I think that ChatGPT has made great improvements over the time. But when I was doing this, it was making hallucinations, right? And I had to go back and fact check everything over and over again, and then rephrase my questions, and it turned into a big cycle. So I was like, is there a way that I can incorporate fact checking into this? And so this is a big limitation to some of the current AI that is out there. And on specific queries where it doesn't know the answer, right, it'll hallucinate up to like 37% of the time. And this is decreased from last year, where it was about like 60%. So compounding this issue is the absence of this built-in fact-checking verification, which means that ChatGPT doesn't cross-check information against sources online, and it can give you false information. And so it's like, okay, I'm tired of wasting my time doing this over and over again. And during this period last year, 2024, a lot of bacterial tools have been released. And when I'm looking at specific newer technologies, right, that's something that at the time ChatGPT wasn't providing me. I wanted things that are released every month with these tools. Different changes are happening very quickly in the space of bacterial analysis. And so I was getting lazy and tired of having these 40 tabs open and adding things to a Google Sheet. And so I said, is it possible to really develop a software that searches key bioinformatics and computational biology packages to provide up-to-date and standard accurate information on currently available software? And so this took a lot of querying. And so I set up some parameters. It's set up so that you can put in keywords that are associated with the software that you want to look for. In my case, I was looking for things like bacteria, microbiome. And if you want to do it with particular like single cell sequencing, you can put in particular phrases or keywords. It associates and makes different tag patterns associated with different words. So for example, you can have microbiome, microbiomes, or microbial, things like that. And then I query CRAN tags. I queried CRAN by different tags that are associated with these keywords, and I queried Bioconductor by these also tags. And I get a list of R tools that are actually on the CRAN website on Bioconductor that are available, right? And so I know that they exist. And then I go to GitHub and I run a separate query over there. And so I break it up into different years because GitHub has like limitations, right, for search queries. And then I perform this search and I retrieve tool and metadata that are associated with it. So things like on GitHub, it's like, you know, how many stars are associated with a particular tool, which is a reference for like how well a tool is used or how often it's used or how popular it is. The different authors, the different citations, when a tool was last updated and things like that. And so I bind all of these together and remove the duplicates. And so for me, it was helpful because I was able to get an outlook that was like, okay, I don't have to go manually searching all these different websites. I can just run it every week or so. It'll provide me newer tools and it'll provide me a summary so that I can put into my dissertation for later or papers that I'm writing. And so that was very helpful. And I was like, yeah, people can use this for other things. It doesn't have to be just for biology or bioinformatics, right? If you want to search software, you put in words similar to like how you do with Google, and this will pop it out. To be honest, it did what I needed it to do. So I'm like not focused on it anymore. But if people want to focus on it, then that's good and find it helpful. And so from this, some of the tools that I had and I would easily come up with, it would provide me like links to the tools, tutorials, the languages that are in, and the citations for me to put in, and different summaries. And so with sequencing populations of these bacteria, right, we talked about how some bacteria are difficult to culture in labs. These steps can take time to grow specific bacteria. TB takes a really long time. And then these isolates don't really represent an entire diversity present. And so we're able to skip some of these steps in order to make sequencing a whole lot easier and analyzing these bacteria a whole lot easier. And so there's been a growth in this idea of population sequencing or plate sweeps. And so this tracks and sour mash, for example, this workflow begins with each metagenomic sample as a raw sequencing read being aligned independently to a set of reference genomes identified by this hash-based search tool, sour mash. And so within this, sour mash really plays a key role in tracks by efficiently identifying relevant reference genomes that match species and strain contents within each metagenomic sample using a mini hash sketching approach. And so this process within tracks really like rapidly narrows down the reference database to genomes that are most representative of the diversity present within the reads, really enabling tracks to align the full read set directly to these references without costly assembly or read binning. And then you have these SNP distances that are calculated rapidly, right? With an IUPAC-aware bit set algorithm. And so it really excludes these high SNP density regions that are caused by recombination. And then you can have this option of using an enhanced trans cluster algorithm. And so it's an enhanced version because it incorporates sampling date information for when samples are collected, and when estimating genetic distances and transmission probabilities between samples, this information is important. And so this temporal integration really allows tracks to better estimate the expected number of not only strains that exist, but the intermediate hosts that really separate pairs of samples and aiding questions about transmission events. And so the final step really looks at clustering samples by the single linkage hierarchical clustering, which is basically a method that starts with each sample as its own cluster, and then it merges the closest pairs based on their genetic distances, and it constructs this like dendrogram or a tree-like structure. And then you use these inferred transmission thresholds to really help to infer transmission pairs that present recent transmissions. And so close genetic relationships are just basically smaller SNP distances in transmission pairs, whereas greater differences represent further away or not related genetic relationships. And so with this, they actually did this on a cohort from the UK called the Baby Biome Study. And so this was mothers and babies, over 100 mothers and over 1,000 infants. And their question was really, how do bacterial strains, how are they transmitted between mothers and their infants? So a very short, right, transmission network when you're thinking about mother directly to the newborn baby neonates. And it showed that the persistence of this beneficial bacteria in infants was happening over time from the mothers to the infants. And it's something that really wasn't able to capture it before, because the bacteria were at such a low abundance that when you have isolates, they weren't being captured. Does that make sense? And so it really demonstrated this reduced strain sharing and it also showed that reduced strain sharing with C-sections as compared to vaginal births. And this really species-dependent persistence. And so it really highlighted the importance of precise strain level tracking for understanding the microbiome, implications for human health, and how bacteria are transferred from person to person. And so with this, I was like, okay, what if we apply different population sequencing tools to larger transmission networks? And so I specifically work with nursing homes and thinking about how patients interact with healthcare workers and the environments around them. Are patients transferring bacteria to the healthcare workers or their environments when they move throughout a healthcare facility? Are they transferring it to workout equipment, for example? And then is it being picked up by a healthcare worker and going back and forth? And so this study is going on like since 2021, and it's still going on currently, and it involves hospitals in Cleveland, Ann Arbor, and Detroit, the nursing homes just down the streets. And it has about 200 patients, and typically these are older individuals which have multiple comorbidities. And so they have about 2,000 surveillance swabs, right? Or just the ones that you go on people's noses, on their body parts, and throughout different environments. And then you also have about 200 therapy appointments. So whether people are going throughout radiology or physical therapy or things like that. And really, nursing homes are a good setting because they're hotspots for disease transmission, right? People are going in and out of hospitals to nursing homes, to ICUs, and back and forth. And so how this looks like is people within these nursing homes can spend a substantial amount of time outside of their rooms for visits, for appointments and for diagnostics. And so how common and important is the transmission as a result of these visits was the question that the original study wanted to answer. And so from this, if you're tracking throughout appointments and following everyone around, this is a lot of swabs being taken, right, of a person's hands when they first start an appointment, after they're using different equipment, and after the appointments, the healthcare workers that they come in contact with. A lot of swabs adds up over time. but it's currently one of the better ways to look at transmission, right? Because you can't use one isolate, and people say, hey, maybe you can use 100 isolates and be able to piece the puzzle together. And so I was looking at how well does population sequencing capture the diversity observed in individual bacterial isolates, right? From the same patient and time points, and what insights does this provide into intra-host bacterial diversity? So instead of picking individual isolates to really represent bacterial diversity, I'm saying, what if we sequence all of the strains of a particular species at once? And so I really want to track how the diversity changes over different sites, if it does, at the same time point. And so when thinking about this cross-sectional, right, analysis of population sequencing, I really wanted to say, hey, the problem is that this sequencing bacterial isolates can really miss much of the genetic diversity. You have to take a lot of them to be able to build a bigger picture. And it only analyzes a limited number of individual isolates. So potentially we can use population sequencing to really get a bigger picture. And the results we were able to capture, right, isolates within bacterial population samples, even those of low abundance. And so typically if you have a bacterial population, let's just say the red circles are low abundance, and you just by chances, right, as you're going through culturing process, you end up not capturing it in an isolate as you end up pick up for sequencing. But if you're looking at population sequencing, we have everything at once, and luckily the resolution is so good that we're able to see things that are in low abundance as compared to just with isolate sequencing. And so this over here is an example of this patient. I tried to shut it smaller. And so different samples were taken across their environment, the curtain, their groin, and the table, and their hands during this day. And with traditional methods, I said, okay, after having all 4 of these isolates, I'm able to separate them down into 2 categories by looking at sniff distance thresholds. For this is Enterococcus faecium. And then the star represents the sample where I have a population sample. And so from that one population sample, I don't need 4 isolates. I can capture the 2 different strains that are there, right? 12 and 13. Without doing all the different isolates. And so really this population sequencing in contrast to isolate sequencing really saves money in terms of you don't have to do as many capturing or surveillance in the hospital taking samples. You don't have to do as much culturing steps and things like that. It's just about how do you delineate it computationally. And so we can even look at things over a longer period of time. And so this patient, for example, has about 15 isolate samples, which is a lot to collect, right? You're thinking about a patient that has to get swabbed all the time, has to remove clothing for the groin and things like that can become a real hassle. And so for the isolate samples that you can see a shift in the strains that are being captured from yellow to blue, right? And so it takes about, 15 samples to see a shift, isolate samples to see a shift in the strains over time. And again, shift in the strains over time is important because it can impact in how we treat different strains of bacteria, right? It can impact the treatment plan. It can impact the overall what's happening in the hospital, right? When different strains go in and out seasonally and things like that. And so what if we didn't have to take 15 isolate samples? What if we took less population samples, right? And so for this one, I wasn't able to pick all the population samples that I wanted, but 4 population samples were chosen, right? The groin, the nares from this initial day. And what I first showed you with bit condensed was just this time point on a specific day in their patient room as a follow-up. And so that was really looking at how one population sample can reveal the strains across one different time point. But now longitudinally, I have these population samples from the beginning of about April until about a month later in the end of May. And what's really cool is that with isolates, you cannot see a shift in alleles, right, for these different strains. But with population sequencing and just having, for example, one sample from the nares and one sample later on down the road for the hands, we can see the shift and changes over time. And so far, able to capture all the isolates in these populations and none of them are missing, is a good step. And I think there's still a lot to do. But this says something about the utility of using population sequencing as compared to isolate sequencing. And so my next step for that actually is to go back. And then my next question is, what about strains that are maybe, how do you know something's not there that should be there? Right now I confirmed that the strains that we're seeing are in the populations, but what about strains that we're not seeing, right? And so how do I delineate those? And so population sequencing, as opposed to isolate sequencing, can be used for tracking strains, right? Because it captures this full genetic diversity within a sample. And it allows for more comprehensive comparisons. It also eliminates some of the biases of culturing single isolates or relying on selected reference genomes. And I think that a lot of tools right now are dominated by, eukaryote analysis. I think that the shift in how we apply tools for bacterial analysis is happening very quickly. And I'm excited to see where it goes. That's it. Thank you.